Welcome to the TOEFL Listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor, or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Hernandez. I wanted to use our office hour to go over CRISPR gene editing applications for my presentation next week.
Good afternoon, Mark. That sounds good. Are you focusing on medical uses, agricultural uses, or both?
Both. The assignment asks for examples in medicine and agriculture and a discussion of delivery methods and risks.
Excellent. Start by distinguishing ex vivo and in vivo applications, because that affects delivery strategies and safety considerations.
Right. Ex vivo is when cells are edited outside the body and then returned. The sickle cell trials where hematopoietic stem cells are edited were mentioned in our readings.
Exactly. In those trials, CRISPR is used to disrupt an enhancer of the BCL11A gene in stem cells. That reactivates fetal hemoglobin and reduces sickling. It is a practical illustration of ex vivo therapy.
And in vivo would be like delivering CRISPR directly to tissues using viral vectors for a disease affecting the eye or liver, correct?
Yes, AAV vectors have been used in a Leber congenital amaurosis trial. That example is useful because retinal cells are accessible, and the eye is immune-privileged, which lowers systemic exposure.
I read about lipid nanoparticles too. Are they being used clinically?
They are. Lipid nanoparticles were adapted for CRISPR delivery to the liver in experimental therapies. They can carry Cas9 mRNA and guide RNA for transient editing, which reduces long-term expression of the nuclease.
Transient expression helps reduce off-target effects, right?
Correct. Off-target effects are a major safety concern. Newer approaches like delivering Cas9 as a ribonucleoprotein complex give the nuclease a short window to act, which lowers unintended cuts.
You mentioned new approaches. Should I include base editing and prime editing as part of the applications section?
Yes, include them. Base editors and prime editors allow precise single nucleotide changes without creating double-strand breaks. That expands CRISPR applications to correct point mutations responsible for many monogenic diseases.
Could you give a concrete medical example to present?
Sure. Besides sickle cell, there are experimental approaches for transthyretin amyloidosis, where in vivo editing reduces production of the misfolded protein. Also, researchers are exploring CRISPR to edit T cells in cancer immunotherapy, to knock out PD-1 and enhance anti-tumor responses.
The cancer immunotherapy example seems broader. How many clinical applications are actually in trials?
Several dozen early phase trials exist. Most are phase 1 or 2 and test safety and feasibility, rather than large-scale efficacy.
Moving to agriculture, what are the best examples of CRISPR application in crops?
There are multiple examples. Scientists have used CRISPR to create rice with improved yield and drought tolerance. They have edited wheat to confer resistance to fungal pathogens, and produced non-browning mushrooms by disrupting a polyphenol oxidase gene.
I also heard about removing allergens from foods.
Yes, researchers have used CRISPR to knock out allergen-encoding genes in peanuts and to reduce gluten content in wheat. Those are important from a public health perspective.
How do scientists edit plants exactly?
Common methods include agrobacterium-mediated transformation, which integrates DNA into plant genomes, and particle bombardment, where DNA or ribonucleoprotein complexes are shot into cells. In some cases, direct delivery of CRISPR MPs avoids stable transgene integration, which affects regulatory status.
You mentioned regulatory status. Does avoiding transgenes make crops less regulated?
It depends on the country. Some regulatory agencies differentiate between transgenic modifications and edits that mimic natural mutations. But most regulators require rigorous risk assessment and field trials, even if no foreign DNA remains.
What about ecological risks for agricultural applications?
Gene flow to wild relatives, unintended effects on non-target species, and loss of genetic diversity are real concerns. That is why containment strategies and ecological monitoring are essential in field studies.
And gene drives were brought up in class as a way to modify wild populations.
Yes, gene drives use CRISPR to bias inheritance and can spread a trait through a population. They are being explored to reduce malaria by changing mosquito fertility or resistance. But gene drives pose high ecological risk and raise ethical and governance questions.
So for my presentation, I should highlight specific case studies and balance benefits and risks.
Exactly. Use sickle cell ex vivo editing and a Leber congenital amaurosis in vivo trial as medical cases. For agriculture, use non-browning mushrooms and drought-tolerant rice. Then discuss delivery, off-target effects, regulatory differences, and gene drives as a public health example.
Do you think I should mention social and ethical concerns too?
Yes, include a brief section. Mention consent in human germline contexts, potential inequality of access to therapies, and ecological justice with gene drives. That shows you understand the broader implications of applying CRISPR technologies.
One last question. How should I explain off-target effects in simple terms for the audience?
Say that CRISPR targets a specific DNA sequence, but similar sequences elsewhere can be cut accidentally. That can disrupt other genes and lead to unexpected outcomes. Emphasize that newer editing formats and careful design reduce that risk.
Thank you. I will structure the talk with 2 medical and 2 agricultural case studies, and a final discussion on delivery and ethics.
Good plan. If you want, we can review your slides before the presentation, and I can point out any technical inaccuracies.
I will email you a draft. Thanks for the guidance, Dr. Hernandez.
You are welcome, Mark. Good luck.
Listen again to part of the conversation. Then answer the question.
Gene drives use CRISPR to bias inheritance and can spread a trait through a population.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.